So our next speaker before the break is Joshua Moore from University of Dundee, who will be talking about scalable format strategies for interoperable bioimaging data.
Hello, everyone. Thank you for having me. And a special thanks to JP. He set this up pretty well. So I really like his um, four types of solutions to dealing with the cloud. And I'll actually be talking about two other possible solutions. But I'm really interested in how we can bring this all together. So, can we make some of these solutions work? Also, I'll start with an apology for this title. It has nothing to do with HDF5. When Andy reached out and suggested I present, I was in the middle of working on a preprint, so the preprint is here. It does talk about HDF5, but hopefully by the time I get to the end of this, I'll have explained what is OME, what's NGFF, and what does it have to do with HDF5. So that's what we're going to try to do. So this project OME, it stands for Open Microscopy Environment. We're a group of research software engineers. We've been around for about 2 decades at this point, and it's our job to provide infrastructure for the bioimaging sciences. So that we provide open source software. We also provide specifications, data resources. We're starting to build data lakes. So it's fairly relevant. All these topics sound quite similar. The bioimaging domain for us is fairly wide, so it covers a number of modalities, primarily light microscopy and electron microscopy, but it kind of bleeds over into some related domains. So neuroimaging tends to be separate, medical imaging tend to be separate, but they do have the same set of problems that we're all trying to solve. For the modalities that we cover, we define a 5-dimensional data model, which allows applications to work together. And that's really our focus. But what's interesting in our space is that there's no standard file format. So I always chalk this up to the fact that building a microscope relative to a collider or something is fairly cheap. So there are a number of commercial vendors, and they produce hundreds of different file formats. And we write a library called BioFormats that tries to read all of these into the standard data model. Over the years, we have tried to propose a file format. So we have one. It's called OME-TIFF. In the bioimaging space, as in many imaging modalities, TIFF is pretty ubiquitous. But it's a 2D file format. So most vendors are actually producing stacks of 2D files that you have to keep track of. And you can imagine as you try to build up 3D volumes out of many individual 2D TIFFs, things can start to slow down. So a number of years ago, we did propose to the community, could we use HDF5 as a standard file format? There are a few vendors who have done this. I mentioned them in the chat. So actually the chat started to move on to the biological sciences during the previous presentation. And from my point of view, when I first saw the HDF API, I realized this is how I want to access data. I no longer want to deal with binary streams. I want some level of abstraction that gives me all of that, that gives me in-dimensional data sets that I can work with. We've never been able to convince our community to make the jump. So we try to work via consensus, and there's always been a barrier. Probably one of the largest ones is the issues with writing HDF5 files in parallel. I know there have been improvements to that, and one vendor, Amaris especially, has been involved with newer versions of HDF5, and they've sped things up. But this is an issue that it's hard to go back and now convince people. So while we were trying to do this and talking to our community about different file formats, actually several members of the community themselves went off and wrote these next generation file formats, NGFFs, which is what I want to talk about. Two of them are quite similar, developed at about the same time. One is ZAR, more in the Python community, and then there's N5 that developed in the Java community. They provide the same basic data model, so similar to what JP is talking about. It's very much a subset of HDF5, but it gets a lot of what people need. And it was written explicitly to live in the cloud, to be cloud native, to allow parallel reading, parallel writing, with no locking necessary. So what we've done is we've stepped in and tried to help the community unify these formats. They're so simple that it's very easy to create more of them, but that's the last thing we want, as from the OME point of view, is yet more file formats. So we're trying our best to reduce it down to a limited number. The use case that they're that they're experiencing here is one that's serverless. So the data is sitting in S3, similar to what JB is talking about. There's no server architecture here. So there's nothing that you can use to rewrite your method calls. And the two options that we really saw are kind of the monolithic upload the file to the cloud and let your users download it. But in worst case, that's gigabytes, if not terabytes of a download. And so just no one uses the data. And these next generation file formats take a very different approach, which is to break up the file into many individual pieces. Each chunk in the HDF5 sense is a separate object in object storage, and then the browser can access those directly. So each URL is completely transparent. And so that kind of gives you the feature set that these communities have agreed upon and are really looking for. So it's a very simple format, easy to re-implement, hacker-friendly, transparent URLs that function identically whether or not the data is local or remote. So, and all of that without any server architecture. And what we've seen then is the community take this idea and build multiple tools on top of it. So this is an application called Visar. It's built on top of Viv, which is a library, a graphical library written in DECGL from the Gellenberg Lab and Harvard Medical School. And we were looking to publish this data. So this is a SARS-CoV-2 data set that we published about a year ago now. And we just put it online, and this group of developers reworked their architecture, their application to talk to this ZAR file, and you're able to publish just with a simple URL. And then what we're working with the community to do is to define more and more advanced specifications. So here we're looking at high content screening data, so many hundreds of images, each of which is as complicated as the previous image, but then look at them at one time in the app and in the browser. And these more kind of advanced use cases then will get submitted back to the VisGL project, so then others can use it. But in our sense, trying to make the domain applications as tangible and as user-friendly as possible. We're doing this for other application environments. So the two that are really big in the bioimaging space at the moment is Nupari from the Python community and Fiji or ImageJ in the Java community. But the feature set that we're enabling is really the same, all based on this specification. But what my dream would be, so what I would really like to achieve, and I guess why I'm here, is a way to bring all these file formats back together again. So the use case I've presented at the HDF meeting would be converting from HDF5 into ZAR to put it on S3. That's what we currently tell our users to do. But we very much think that if the metadata is equivalent between the different file formats, that actually the user need not necessarily know which of these formats they're using. Functionally, they can all be identical. And our hope is by having a small number of formats that we're providing to commercial vendors, that there will no longer be new proprietary file formats. So we hope that we've now found kind of the magical trio that will make everyone happy. And to the extent that we can make HDF5 and ZAR work better together, I'm all ears. So that's certainly what I would like to discuss. So I have a few minutes left. I'm going to talk about one particular way of making them work together. I'm not necessarily saying it's the best way, but just to kind of get in the details of what this looks like. As I mentioned, so we're currently using a conversion workflow. The tool, as I mentioned, that we build is called BioFormats. So here you can run BioFormats, and you can take this OMERO, which is a HDF5 based format, and convert it into a ZAR. And you get on disk a large number of files, right? I mean, I think for the largest image I have, which comes in at about 5 terabytes, that's 14 million files on disk. And that's not great for a POSIX file system, but it functions. But these local files are also the URLs that you'll be publishing onto S3. So the attributes at the top of the orange box contain all the metadata necessary for interpreting the image, and then each of the data sets, number tier 0, 1, 2, and 3, are the different levels of a multi-resolution pyramid. So this is really the entire thing that we're trying to publish. A recent project called FS Spec Reference Maker, we need to work on a new name, has begun. FS Spec is a library in the Python space which allows kind of a transparency of backend storage. The user doesn't need to know whether they're talking to S3 or HTTP or GSS or a local file system. They just give a URI accessing the data. But whatever backend you're talking to, you request given keys. So in the previous example, you request the o.o.o.o file and you get it back. But there's nothing that will let you look into a single file to take out the individual chunks, i.e. there's no way to work with HDF5. So FS Spec Reference Maker introduces a redirection layer, which means you can take a HDF5 file, you can generate this JSON redirection layer, which looks something like this. And then as the user comes and asks for given ZAR URLs, so here, for example, the attributes, those values can either be inlined into the JSON if they're fairly small, so you can just have it all immediately in the JSON, or more importantly, if you're looking for a binary chunk and you can't put all of that in JSON, you just give the offset and the length of how to access that at the given URL. So this is saying, go to S3, look in the IMS HDF5 file, go to offset 340,000 and then read for 40,000 bytes, right? And that allows us to provide ZAR URLs on top of a HDF5 file. Which means in the ecosystem, I won't go through all of this, but this is kind of the ecosystem that was evolving in bioimaging for using these ZAR files. Have them remote, have them local, but always telling the user to do an upfront conversion. This is one of, I think, JP's method 3 of working with S3. But then the question becomes, could we keep the HDF5 files and the TIFF files? The same thing works for TIFF files. And by adding one of these index files, which I think was option 2 in JP's presentation, could we make them accessible via the ZAR API and kind of have a consistent way of accessing all the data for all of our users, which, yeah, consistency is a good thing. So those are the ideas around how to make this happen. I went fast. Hopefully it wasn't too fast. I'll just show the acknowledgement slide. I won't go through it. Obviously, lots of people involved in making this happen. But yeah, interested to hear what you have to say. Thanks.
Thank you, Joshua. Any questions?
I did have one question, but I don't know if I should just go to the website. But N5, anyway, thanks a lot, Josh. It's very interesting and it challenges our own models. So it's very good to be challenged of only HDF5. But what is N5? Is N5 similar to what you Matt just presented?
No, it's a good question. N5 stands for not HDF5. It's a bit tongue-in-cheek in the sense of like no SQL. So it was developed at Janelia by the Stefan Saalfeld lab. It looks almost identical to ZAR. So what we've done is we actually got funding from the Chen Zuckerberg initiative to work on bringing those two specifications together. There are a small handful of other formats like this. Some use YAML, some use JSON. It's pretty easy to just put binary on disk and put a little metadata. But the community realizes, for the longevity of any new format, you need, long-term support and you need a proper specification and kind of all of these things. So there's definitely a willingness to work together, but I think the take-home that we received is there needs to be something like this. There needs to be this kind of simple option, and that's what we are in the process of looking for.
And Mark, please go ahead.
Thank you. Thanks very much for the presentation, Joshua. I was really interested in the slide you showed and the description around wanting to see commonality of metadata between ZAR implementation and an HDF implementation. And I just wanted to mention that I've been involved in some research work quite recently looking at one of the HDF derivatives and looking at extracting metadata graphs from files for indexing purposes, but also thinking about being able to recast that metadata into other formats. And I think being able to see a sort of separation of concerns between the record, which is the descriptive metadata and how that's structured, and the encoding of the very large volumes of data on disk has some real value. And yeah, I might be really interested to follow up with that further with you.
Yeah, definitely. I mean, you said sorry to ask questions in the other direction. You said graph. Anything in particular? So I'm a RDF fan. So
yeah, we've been building RDF graphs out of HDF and NetCDF metadata.
Okay, wonderful. That's exactly what I want to do. So yeah, definitely let's get in touch.
There was a question.
Okay, I will sling you an email and we can discuss offline. Thank you very much.
Thank you. Sorry. There was a question about if there is any HDF5 wall implementation that allows to store chunks in the same way as ZAR storage directory store does. At this point, there is no wall implementation. We would like to have it, so we have direct access to so-called HDF5 optimized storage, which is very similar to ZAR format and what was presented here in this talk. John Reedy will be talking more about HDF5 optimized class format, but the idea is the same. Each chunk of data is stored as a separate object, and metadata describing data set is stored as a separate object. And it is possible to write wall connector that will be accessed directly that storage, and it can be implemented locally too. So stay tuned for tomorrow's talk, and we agree that it will be to have serverless access to HDF data will be great. And by the way, you do not need to chop the file. You may now access it in a S3 directly to the file when you know location is a chunk, as you are doing.
So just for everyone's benefit, unfortunately, I won't be able to attend tomorrow. I have a conflict, but I would very much like to continue that conversation as well. You know, I can think, going back to Andy's question about N5, the N5 community wrote their own HDF5 backend. As crazy as that sounds, the not HDF5 library has an HDF5 backend as well, you know. And so I assume the same would be true for ZAR. You know, it makes complete sense to have a HDF5 backend for ZAR. And then, you know, obviously, I guess where I'm going is it would also be interesting to have a ZAR backend for HDF5. You know, can we make all these things just completely transparent and work together? It would be amazing.
Yes, thank you.